Happy Sunday, everyone. This is your punctibyte for the week of February 17th. It's tough to believe that time has passed so quickly. I hope you all had a great weekend. We had our home race this weekend, and it was a huge success, although today was incredibly harrowing with waxing conditions, trying to wax 30 some odd skiers in very hot conditions. So ask me about it sometime and I'll tell you more. It was kind of a sloppy mess. So I wanted to hop in with a few things that maybe will give you a little bit of background for the week. First off, obviously scheduling is a big must. So let's talk about what we're going to have going on this week. Lab 8 and 9 for most of you. Erin and Aaron will hop along to Lab 10 and 11. So that's a big day for the two of you. We'll talk just a little bit about that. Caleb and Mitchell, you're going to be on for Open Lab this week. So trying to get somebody to either cover you or to be there for your Open Lab would be absolutely marvelous. So we'll talk about the TA guidelines for both Lab 8 and 9, but the first thing that I want to do is actually to talk just a little bit about two things. One, a quick Canvas update, and that is for some of you, and I think Jesse was amongst this group. A few of you have asked me about doing mobile and being able to work with Canvas on your mobile device, whatever it might be. And so I have my iPad with me today, and I'm going to show you how to go about that. See if we can be tricky here and have the iPad and the computer screen going at once. But basically, this is what you'll want to do is you'll want to search for your app store. So basically, just going to your apps. The confusing thing is once you get to your app store, what you want to do is you want to search for SpeedGrader rather than searching for Canvas or YO Coursers. So once you can find SpeedGrader, you should be in good shape. And SpeedGrader will allow you to grade most everything with this cool app. So this is the name of this is the look of the screen once you bring up SpeedGrader, and you can go ahead and hit on this icon and it will allow you to grade anything that you have in the gradebook. So maybe you put some some sort of fill in the blank question or something of that nature on your pre-lab, and you want to be able to grade on your pre-lab. So you just click on open there, and then you can go ahead and grade anything that says that it's in need of grading. This is obviously not exactly what you guys would see because you would see your courses, but once you're there, it will show you, just as it does in the regular Canvas, what you need to grade. So this is a cool app. The other thing that I wanted to show you is an article that talks about Pseudomonas aeruginosa, and this I thought was a good time to talk about this article because this coming week we will be talking in Lab 8 about Pseudomonas and Staph aureus and talking a little bit perhaps about their sort of clinical pertinence. So this particular article is an article about Pseudomonas has a type 3 secretion factor that is very important in its virulence. And this article just quite literally shows that if you make a strain of Pseudomonas that lacks the ability to use this particular secretion apparatus, it actually is completely avirulent. So it's very much an important part of its ability to cause disease. So remember that this week in Lab 8 we will be talking about antibiotics and antibiotic resistance, and looking at Pseudomonas as it grows on a Kirby-Bauer plate. We'll notice that it does secrete piocyanin, and this will be the student's best look at piocyanin. So for Joe and Callie, I usually try as a supervisor to talk about both Staph aureus, about resistant strains of Staph aureus, methicillin resistant or MRSA. I also like to talk about Pseudomonas as a cause of nosocomial infections and its secretion of piocyanin as a virulence factor. So if you also want to shoot in there that much of Pseudomonas' virulence can come from things like quorum sensing, but also also obviously from secretion apparati. So kind of cool to give that as a segue into our conversation about Lab 8. So your TA guidelines for Lab 8 give you a lot of background on the just basic lecture, where we're looking at things like UV light, the Kirby-Bauer, and resistance selection for streptomycin resistant mutants. A lot going on this day in the lab, so it really makes a lot of sense to split students into 3 groups, where one group goes and does the UV light station, one group does the Kirby-Bauer, one does streptomycin resistance, and then that way you don't ever get crowding at the UV light box. Try to get them encouraged to do really fun foil shapes, and maybe, you know, Valentine's Day being just recently, they'll want to do star or hearts or something like that. But whatever they do, make sure that they do take the plate lid off before they expose it to the UV light. So recognize that if you have somebody back at the UV light station, you might actually stop problems with exposure, but you are in both ways, I guess. You'll stop problems with underexposing the plate by not taking the lid off, but you'll also stop problems by stopping people from exposing themselves, and sticking their hand under the light or turning it around and looking into it, which is sort of funny for us, but it's not so funny for them. So definitely get somebody back by that UV light station. Also, in terms of the Kirby-Bauer assay, we have some students who are allergic to penicillin. Some of you may be having students who are not coming to lab. I always err on the side of safety with that, so I tell them not to come if they have any kind of severe allergy to penicillin at all. But if they do come anyway and they say, oh, it's a minor allergy, please have their lab partners be the people who do the majority of the work at the Kirby-Bauer station. In terms of streptomycin resistance, this is one of the hardest experiments that we do conceptually, and the reason for that is because they're doing the same experiment twice. So they're going to be both selecting for E. coli mutants in broth and in agar. So they'll be putting streptomycin into agar in one plate, but not into the other plate, and they'll put bacteria into both. Same thing in broth. They'll put streptomycin into one broth and not into the other broth, and so we'll be able to select in one tube for the resistant mutants, or vice versa. In one plate, we'll be able to see the resistant mutants. We have put TTC dye in the one plate or into both a plate so that the color of the growth, the bacterial growth, should turn red. That doesn't always pan out perfectly, and sometimes we just simply do not see good isolated colonies on the resistance plate. So we'll just have to play it by ear and see how it goes. Lab 9 is coming up on Thursday, and then for most of you, Wednesday or Thursday. And for Lab 9, we'll be continuing our theme of antimicrobial components or antimicrobial substances. So antiseptics and disinfectants. So this is another note. If we would go ahead and put on the board during Lab 8 that they can bring an antiseptic or disinfectant from home, that would be marvelous, because they can test whatever antiseptic or disinfectant that is as long as it's in liquid form during Lab 9. So Lab 9 is actually one of our most straightforward labs in terms of the lecture, really just talking about sterilization versus sanitation, and about the differences between antiseptics and disinfectants, and by definition what they do differently. It's a really fun day to grab aside the sporicidin, which is the old 1950s stuff that claimed to kill 100% of all organisms on one side, and then on the other side it says won't yellow hands. So it's very comforting to know that something that kills 100% of all organisms won't yellow your hands. But it's kind of fun to pass that around and to talk to the students about that. In terms of the hand washing experiment, please do split the students into 2 groups, or maybe 3 groups. You could have one group that does the hand sanitizer, one group that just washes their hands, and maybe you could switch it up maybe between 2 types of hand sanitizers. Whatever you would like to do for those groups, just make sure you make it clear to your students. We also follow up during Lab 9 on the streptomycin resistance isolation. It's very important to show them how to take the plug with the isolated colony out of the previous plate and to resuspend it in the saline solution. That is a tricky technique because they need to put their pipette down over the plate, and then they indirectly with the bulb down, and then they need to turn it sideways and let the air come in and suck up the plug kind of on the side. So that'll pull up the plug into the sterile pipette. You may need to demo this for them. I will try to do that in some of the sections that I'm in, but if I forget, please do help me remember. Let's lastly cover Lab 10 and 11 and the TA guidelines for these. I often give this lecture. However, on Tuesday of the week that I'll be doing it in my section will also be an exam week, so I would love for some of you to get jazzed about doing this and really doing a good job of it. We have talked a little bit about lateral gene transfer in lecture, but we haven't gotten to the point of the full coverage of recombination. So we're not the students don't have full background, though they are pretty jazzed about bacterial cell sex, and you can't blame them, but that's not something that we've given them the full lowdown on. And I think it's great if we can help them understand the difference between mutation and recombination. And remember that with mutation, that's a change in an organism's own internal DNA, whereas recombination is a recombining to make a new genotype in which there's DNA from a donor cell that is transferred to that recipient cell. So there's DNA from two organisms in recombination, and that's a very important differentiating feature between the two. Logistically, Lab 10 is difficult. We have to have them start right away. As long as we have them work through steps 1 through 6 prior to beginning anything else, then everything is fine. When they hit that 20-minute incubation on ice, that's when we hop in and give the lecture. And some of you will fit the lecture into that 20 minutes. I usually am pretty wordy and I go over that 20 minutes, so I'll pause the lecture and I'll say, now go heat shock your cells, put them in the shock, then put them in the recovery buffer, and then put them back into the 37-degree E. coli spa to recover from the shock. And then I go and I finish the rest of the lecture during the 30-minute incubation. Also, during that 30-minute incubation, we need to have them work on their results from the time before, looking at their antiseptics and disinfectants. We also need to have them do their Lab 11 procedure. This is just a prep step for Lab 12, but I just haven't renumbered it, and so just have them do this prep step. All it is making a lawn of E. coli with a hockey stick and then spotting filtered sewage onto 3 of the 4 quadrants, with the 4th being a control with sterile broth. So that'll be done during that 30-minute incubation as well. Finally, as students are waiting to plate their recovering E. coli, have them go ahead and write the predictions for how the results will turn out for their transformation procedure. So if they can do that before they leave, that's the key to you as to whether or not they are fully understanding the concepts that we covered as far as which of the genes will be inducible and which are constitutive. And so those concepts will come through in those predictions. Okay, I think that wraps up and sums up what we need to have under our belt for the week to come, and I'm looking forward to seeing all of you. The lectures have really been improving, and this week I'm hoping that I'll be able to bust out my camera and give you a little bit more feedback by showing you your lecture, which should be kind of fun. Okay, have a good rest of the weekend.